Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there. Hello everybody, welcome back to another episode of the A-Level Cookbook. Today we're going to be doing the OCR A-Level Biology A June 2018 page. So let's get started. So which of the following statements correctly explains a feature of an efficient gas exchange surface? So the rules are, when you're doing gas exchange, you want to max out your diffusion. You want a diffusion max. So what that means is you want to increase the surface area to volume ratio as high as you possibly can. You want to keep the concentration gradient as steep as you possibly can. As well as that, what else do we want? We want the shortest diffusion pathway known to man. And as well as that, we also want to have a high temperature, but obviously you can't really control that. You can't just make your temperature go up. So, that one's kind of one, right? But then as well as that, you also want the highest possible amount of like channel proteins or protein, carrier proteins and stuff like that. Because then you're absolutely maxing out the ways of possibly, doing those kinds of things. So whichever options facilitate this is going to be the one that's correct. So if we have a look here then, it says the layers are thin for a short diffusion distance. That is definitely one of them. There is a good supply, so it reaches equilibrium quickly. No, we don't want equilibrium, because if you're reaching equilibrium, what happens, I don't think a lot of people realize, is high to low. As diffusion's happening, the gradient's getting less and less and less and less and less and less and less steep. So you might start at like 100 and 0, and then you get like, 80, 20, so forth, blah, blah, blah. Eventually you get to like 52 and then 48. The gradient's not steep at all, so the rate of diffusion is going to be really slow, and it's going to take ages to actually hit 50 and 50. So that's why we have things like, for example, moving blood away from it, or like, ventilation and stuff to keep the gradient really steep. Otherwise, it would eventually reach equilibrium, and that would stop it. So you're out. There's an increased surface area to reduce the surface area to volume ratio. That is incorrect. You want, if you think about a sheet of ice, right, a very thin sheet of ice. Sorry, whoops. You have a thin sheet of ice versus a cube of ice like this. Which one's going to melt first if you hold it, even though they're the same, length and width and height? Say the same length and width. This one's going to melt first because there's a higher surface area to volume ratio, because it's a high surface area. It's nice and flat, but the volume's not very chunky. So if you held it, the heat can move through it and melt it quite fast, whereas here it's got to go all the way through there. So we want a really, really high surface area to volume ratio. So that one's out. Next it says ventilation takes place to reduce the concentration gradient. No, we want it to be as high as possible, so it's A. Next it says the following are a series of organic molecules and the chemical process that convert them into different ones. Which one is correct? So just got to look at them one by one and have a look. So nucleic acids via hydrolysis makes nucleotides, which hydrolysis polynucleotides. No, because a nucleic acid would join onto a nucleotide, and hydrolysis is breaking it apart immediately, and nucleotides are joined together to make a polynucleotide by condensation, so you're out. Next we have a look at the next one. So it's alpha glucose is condensation reaction into amylopectin. So far so good. Which then if you hydrolyze goes back down to amyloglucose. You break alpha glucose, sorry, that's correct. B. Amino acid condensation can do that, but if you hydrolyze it, it's not going to do that, because that's, remember, breaking down. Beta glucose, if you condensation, you do make cellulose. If condensation, again, it does not make maltose, because maltose is a disaccharide made of alpha glucose, so you're out there. Then it says the table describes the percentage mass of different chemicals in organic polymers. Which one A to D is correct? So if we have a look, we've got nucleic acids and all of this stuff and whatever, right? So it's just a case of kind of remembering, but also figuring out as well, just on some logic. So a nucleic acid is going to be pretty chunky in nitrogen, carbon, oxygen, hydrogen, and phosphate. Literally all of them. The nitrogen particularly. So that's looking like one of them. Carbohydrate, carbon, hydrogen, 8 oxygen. The fact that, there's this, and this is pretty promising. But they're not in the same proportions. C6H12O6 is glucose, for example. It's not just all 1 to 1 to 1 to 1 to 1, so that's out. Protein, nitrogen, carbon, oxygen, hydrogen, phosphorus, sorry. Well, I mean, it's going to have hydrogens in, no. Nitrogen, carbon, oxygen, hydrogen, no, because there's still going to be a lot of hydrogens in there. You don't have that much like you've got your long fatty acid chains with loads and loads of hydrogens on them, so that's that as well. So in this case, it is A. Now it says, which of the statements about DNA replication is correct? So RNA will bind to DNA through complementary base pairing. That is objectively wrong. Why is RNA coming into this when we're doing DNA replication, where you take a DNA strand, you use each one as a template, and then you for the other nucleotides to form complementary base pairs, and then DNA polymerase puts them together like that with a condensation reactions between the nucleotides that are adjacent to each other. RNA is single-stranded, and it's not even relevant here. So why is that here? Get out. The distance between the strands and the double helix will always be the same. That is objectively also not correct, because you've got the coiling and folding and whatever, so nope. The proportion of adenine will always be equal to the amount of guanine. No, because they're not complementary base pairs, so you're out. The formation of the phosphate ester bonds will occur in the same direction in each strand during DNA replication. That is indeed correct, because it works in each direction like that, so it is D. Next it says, the following graph shows the rate of reaction of an enzyme at different substrate concentrations. Which one shows the rate with a fixed quantity of a competitive inhibitor? So if you look here, we're changing the rate sorry, they're changing the concentration and measuring how the rate goes along. So if you put a competitive inhibitor, it kind of binds to the site, then detaches, binds, and so forth, right? So eventually, if you're putting the substrate like really, 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 really high, there's your inhibitor, right? Pretend that's a square, and then there's your substrate. Eventually, you've got so much substrate that the substrate is more likely to collide with the enzyme to more likely to collide with an enzyme, leading to an enzyme substrate complex versus the inhibitor. So eventually, the effect is going to be, you've got so much of it, it's just going to, be back to normal. So it's not A because that's like the highest rate, right? And it levels off here. B is like a slightly slower rate that it's going up with, but it eventually levels off at the same point, which is in keeping with what we want to say. C and D can't be true because it's competitive inhibitors. It's not going to actually change how the enzyme works. In theory, as long as you keep increasing the substrate, eventually, you'll get more likely to have the substrate collide with the enzyme and lead to an enzyme substrate complex versus a non-competitive. Non-competitive would actually change the shape of the enzyme entirely, right? Which means it can't actually reach the same peak even if you max out the substrate because some of them will be screwed up. So that's why these go down. One of these two, anyway. So it's got to be B. Which of the statements A to D apply to transpiration and evaporation? So transpiration is, remember, the loss of water through a leaf, through the stomata when it's evaporating, and evaporating is just changing from liquid to gas. So we need to think about what's relevant to both of these. So it occurs at a faster rate and higher humidity. Transpiration does, but evaporation is just changing from liquid to gas, so no. It occurs at a slower rate at a greater wind speed. Transpiration is faster because you're moving the water away, right? The moment it's being pushed out, eventually diffusing out the stomata as a gas. So then, if you're doing that, the concentration gradient is steeper, so it'd be faster. So you're out. It's at a slower rate at higher temperatures. No, that would be it's objectively not correct because you've got transpiration, more kinetic energy, meaning it's got more kinetic energy to move away and so forth. But also you're evaporating it faster because you're heating it. So that one's out. So it's got to be D, and that is indeed correct because it's through the stomata, through the leaf surfaces, and it's evaporation from the inside and the water leaving out through the stomata by diffusion. Next is which of the statements explain why diastole follows systole in the mammalian heart. So systole is the contraction of the ventricles. Diastole is their relaxation. You can also say atrial systole for contraction and atrial diastole, but a lot of the time when people say systole and diastole, they're talking about the ventricles. So it says which of these explains why this follows this. Cardiac muscle is myogenic doesn't explain why this diastole comes after systole. All that's saying is that it can beat on its own without nerves going to it, so that's out. And it says cardiac muscle takes a short time to repolarize after being stimulated. So if you stimulate cardiac muscle, it's going to contract. If you're contracting, and then here's your friend that's contracting, and then this is your relaxing, it's contracted, it's going to take some time to get back to normal, and then it's chilling like that, right? That's possibly true. That means there's a bit of a gap between the two of them, and when it's repolarizing, it's relaxed. The aorta is capable of maintaining the pressure generated by that. Doesn't explain why diastole comes second. The sinoatrial node receives impulses from the AVN. That's the wrong way around. AVN gets impulses from the sinoatrial node, so that's also out as well. So it has to be B. Banting and Best pioneered experiments in the functions of the pancreas. That sounds like a bread brand, like, malted bloomer, crusty white, Banting and Best. But anyway, in one experiment, they reduced the pancreas to duck shortly after they got symptoms of diabetes. Banting ground up the removed pancreas to produce an extract. They called it iletin, and then they put the iletin into dogs that had their pancreas removed, and then they tested the blood glucose concentration. So the blood glucose concentration, you can see when they're doing this, iletin goes down right to the bottom, and then goes back up, and then down, and back up. And they were like, interesting. And that's also interesting to think about, is like the concentration's high, whatever. When they're injecting it, it's going down, but then why is it going back up again? Hello? What's going on here? Surely it should just stay down. If you think about it, if it's going down, that's the insulin doing its job. If it's going up, it's the glucagon, which also comes from the islets. So then, it says which of these explains the results correctly. So, iletin is made in the alpha cells of the islets of Langerhans. No, it's made in the beta cells. So that one's immediately out. The iletin reduces the blood glucose concentration. Well, most definitely does. And the effects of it are short-lived. So it's clearly a somewhat short-acting thing, and it's coming back up, and they have to keep redoing it to keep it down. So it's definitely that as well. So in this case, it's got to be 2 and 3, which is C. Peak expiratory flow rate is a measure of the maximum rate at which a person can exhale. The graph shows the typical PEF values for men of different ages and heights. So if you've got asthma and you've had to breathe in that tube, that's exactly what this is. So which one of these shows the percentage increase from PEF from a 20-year-old man of this height to a PEF of that height? So what we need to do is we've got to do a couple steps. We've got to look at the axis, so you can see that's the PEF and that's the age. We're looking at a 20-year-old man. So if I look for where 20 is, and I draw a nice straight line going out. So looking at 175 on the 1, 2, 3rd line up, that's what, 570 for the 20? And then for the other geezer, it was 183. That's the second line down. 45 years old. Here, that's 640. So for the 45, it's 640. It's saying what's the percentage increase from the other two. So it's going to be 640 minus 570 over 570 times 100. If we do that, we get 12.28, which is 12.3, so C. There you go. One treatment for thyroid cancer is radioactive iodine, and this isotope is used. The thyroid gland absorbs any iodine that enters the body so that the radioactive iodine kills the cancerous cells in the thyroid gland, and then it's excreted from the body. So different body fluids excrete different proportions of it, as shown as in this graph. So this is the amount of excretion, the rate of it, how fast it's done: urine, feces, sweat, and expired air. Cool. So in this case, now we need to figure out why, for some reason, it's really high in the urine, and it's kind of right in the feces, it's kind of in the sweat, but it's barely any in the air. So whichever one explains this best. So in this case, it says I-131 is very soluble in water. Well, if that was the case, why is there not very much of it in the sweat? Nah. Next it says it can cross capillary walls. All of these would have eventually left capillary walls. Like, capillary walls into the lungs, capillary walls into the skin, and so forth. Capillary walls into your poop, and it just doesn't explain that. The kidneys are more efficient excreting the iodine 131 as the lungs. Well, the lungs would be the air and the urine. That's looking like it's good so far. And the thyroid gland is well supplied with blood. That doesn't explain anything, because it all eventually end up in one of these. So it's got to be C. Which of the following describes the similarity in which ATP is made in respiration and photosynthesis? So in photosynthesis, it's the light-dependent reaction where you have non-cyclic photophosphorylation and also cyclic photophosphorylation. So what happens in the light-dependent reaction is an electron gets very gassed, either from photosystem 2 or 1, and it's because it's shined light and absorbed it and been excited from the chlorophyll, whatever, right? But then it moves down an electron transport chain and it needs to lose energy. So what it does is it uses that energy to pump protons into the stroma like this, and then it goes out through the ATP synthase. Whereas in respiration, it's all during oxidative phosphorylation, where the NAD dashes it to the electron transport chain, and via a similar process, the energy from the electron, because it's too excited, it's like, yo, I'm too happy, bro. I don't want to go anywhere. I'm free. All the energy's gone to pumping H+, same shtick, and the same thing. So whichever one of these makes most sense. So both involve NAD, that's not correct, because plants use NADP. Both involve substrate level phosphorylation. No, not really, because the light-dependent reaction doesn't do that. So no. Both involve photons. No, because we don't need light to do that. Both involve proton gradients is correct, like I just said. Then here we go. It says the diagram shows a human brain. Which ones would directly involve in learning to play a musical instrument? So let's have a look at this brain, shall we? So the gross structure we've got going on here is that this stuff is the cerebrum, the cerebral cortex, right? So the cerebrum and the cerebral cortex basically is all about your voluntary actions, like your moving, personality, things like looking at things and actually making meaning with it and stuff like that, depending on what region you're in. This area is your hypothalamus and your pituitary gland, so the reason they all do stuff to do with, for example, regulating temperature and water balance, and then this is also storing and releasing hormones for all kinds of different bits and bobs. Here, that's W, that's your cerebellum. Cerebellum literally translates to little brain, and it's all to do with things like posture, balance, and movement, and coordination. And then here, X, that is your medulla oblongata, which is all to do with autonomic control, for example, your heart and breathing rate. So it says here, which one's involved in learning to play a musical instrument? Already X is out, because that's all automatic unconscious stuff. Now Y is all to do with hormonal stuff, so that's out and that's out. So all you've got left is C. And that makes sense, because you'd want to be able to coordinate your hands and your movements and stuff and your posture to be able to play the instrument, and Z, you want to be able to actually recall all the information needed for it and actually hear what you're doing and interrogate and make meaning of it. So there you go. Which of these diagrams shows a sensory neuron? So sensory neuron, remember, they have one dendron which carries an impulse towards the cell body, and then one axon which takes it away. So whichever one of these has that in the middle, already that's going to give us this. The other ones, why they aren't correct. So for example here, if we look at A, so dendrons, remember, carry impulses to the cell body, and an axon which is away. Motor neurons, what they have is that they'll have one long axon and lots of dendrites. This is cell body, and it's got lots of dendrites, and it doesn't look very long because they're all kind of short, so that's going to be a motor one. B, that's a relay neuron, because they've got many short axons and dendrons, and they all communicate impulses between sensory and motor neurons, or all kinds of things like that, basically. So that's that. And then here as well, what's going on here? Well, it wouldn't make sense for it to just be like like dendrites and an axon, for example. Like, which way is things going, bro? Like, what is this? What even is this thing, dude? So that's out, so it's D. The table below shows the membrane potentials of different neurons at cholinergic synapse. So remember that synapses is where you have a presynaptic neuron, you've got a postsynaptic, whatever it could be. It could be another nerve, so postsynaptic neuron, or it could be like a muscle or a gland or something. What happens is a cholinergic synapse is you have an action potential arriving here, because there's depolarization of the presynaptic membrane. What happens is that this opens voltage-gated calcium ion channel, Ca2 plus enters here, and Ca2 plus then promotes vesicles containing acetylcholine to fuse with the membrane, and release the acetylcholine into the synaptic cleft. The synaptic cleft, and then it diffuses across. The cleft is a scab, by the way. It will diffuse across, and then it will eventually bind to a complementary receptor here, and then this will open up ligand-gated, so they open when this is, stimulated and something's bonded to it, sodium ion channels. So then you get depolarization here because it's becoming more positive, and if a threshold's met, you get an action potential there, bang. So in this case, we're looking at exactly that. So presynaptic, postsynaptic, and what's going on here. So if I just draw these, keep that drawing there. It says which one of these explains these data. So presynaptic neuron A, presynaptic neuron B, presynaptic neuron C, and then postsynaptic neuron. So its membrane potentials of different neurons and the separate equations are shown in the 5 rows. So presynaptic neuron A was positive, negative, negative, and the postsynaptic was negative. And then for some reason, so we've got multiple here. Right, we've got 3 of them. Oops, that's really badly drawn. Negative, positive, negative, 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 positive, negative, then positive, positive, negative, negative, and then positive, 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 positive. So it seems like this seems to be a culmination of every single one of these is together. Right? And these are all 3 different neurons secreting acetylcholine onto this, causing, an effect. So if it's negative, then what that means is that there's no, there's not been any depolarization, the acetylcholine's not diffused across, blah, 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 right? Whereas if these are positive, they've got an action potential coming through them, so it must be that the acetylcholine is being released, blah, 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 blah. So it is binding to those receptors, and having all 3 of them is enough to make this one become positive, so that one's having an action potential. Whereas when you have only 2, for example, it's not enough. When you have, 1, it's not enough as well, right? So if that's the case, the effect of all of these 3 are added up, so it is spatial summation. Summation is adding up the effects of all 3 of these. Spatial is they're doing it in space, within each other. Temporal is to do with time. So for example, if we only had 1 of these and it kept releasing acetylcholine, and then obviously it gets broken down, blah, 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 right? If this kept releasing really fast amounts of acetylcholine before it's even had time to be taken away, then eventually the generator potential would add up, and then you get the threshold. So this is adding up based on time. This is adding up based on multiple, putting their acetylcholine onto one neuron like that. The drug metoprolol prevents stimulation of the postsynaptic membranes in the sympathetic nervous system. Which of the following conditions could this be used to treat? So with postsynaptic neurons, if it's stopping that happening and it's stopping them being stimulated, it's depolarizing them. Muscle fatigue, it's telling you this is the sympathetic nervous system. Like muscle fatigue is voluntary skeletal muscle, which is not even in the sympathetic nervous system, because your nervous system, right, is divided into your central nervous system, which is your brain and spinal cord, and the peripheral nervous system, which is everything else that is not your brain or spinal cord. This is then divided into your autonomic nervous system and then your somatic. Soma means body, so your body nervous system that you think about and you do. So if I I'm writing this now, I'm using my somatic nervous system. The autonomic nervous system is all your automatic stuff that you don't think about, and that's your sympathetic and your parasympathetic. This is all to do with things that you don't think about, so muscle fatigue is not coming out in this context. It's going to be this. So that one's out already. Tachycardia, tachy, fast heart. Fast heart means high heart rate, so yes, it could do that, because if it stopped sympathetic impulses of that. High blood pressure, same thing here, so it's 2 and 3, which is C. Gestational diabetes is a medical condition that affects pregnant women. It results in high levels of glucose, even though they're producing normal levels of insulin. Which type of diabetes is this most similar to? So type 1 is where you are not producing insulin at all, because your beta cells have been attacked by your immune system, whereas type 2 is where you're still making insulin, but your cells are not responding to it anymore for reasons like, for example, those insulin receptors are damaged. So in this case, they're still making normal levels of insulin. Type 1's out, so it has to be type 2 by default. Type 2, why? Because insulin is still produced, right? So it must be that the insulin receptor may be damaged or something, and it's no, your tissues are not responsive. There you go. So just 2 ways a woman with gestational diabetes can manage their condition. So, I mean, one of them is the same as all the other stuff as well. So, for example, you would lower, for example, carbohydrate intake. Or you could give them, drugs, for example, like metformin or something like that. Or exercise too. Which is kind of rough. Imagine being pregnant and being like, yeah, sorry, mate, you'd have to go do some runs every day. Next it says, skeletal muscle is one of the main tissues where glucose is removed from the blood in response to insulin. What is the other one? It is the liver. One way of remembering that is that the liver is rich in glycogen. So if there's glycogen there, there's glucose being stored, so it must have taken glucose at some point. Explain why glucose is required for the contraction of skeletal muscle. So, I mean, you should, and I hope, are saying that, first of all, it's used in respiration, right? So you're using it in respiration, which makes ATP. Generally speaking, when you're talking about glucose and energy and anything, it's going to be respiration making ATP. So then, it's saying, why is this helpful in skeletal muscle contraction? So think about your just a real quick crash course, think about your muscles. So you've got, how they work, the sliding filament theory. We've got our actin, we've got our myosin chilin here. whatever, right? The myosin globular head. So what you usually have is you have tropomyosin with troponin here, and the calcium binds to this and drags it across like that away when an action potential gets here, and the calcium is released from a sarcoplasmic reticulum, which is where it's all stored. The myosin globular head would then bind here like that, with forming an actin-myosin cross bridge, and then what it will do is it will hinge that way, dragging the actin that way, until it hits another point where there's the next actin-myosin binding site, and the same thing happens. It would bind here, and then it would cross, it would do that. Drag it across, right? Then, once that's happened and it's all done and dusted, we need to detach the myosin and put this all back into place. So we need to break the actin-myosin cross bridge, so it needs ATP to do that, so that then it detaches, the myosin head's chilling here. We need to get rid of the calcium, so we need to actively transport it back into the sarcoplasmic reticulum, which, requires ATP. And then eventually the muscle, the actin that you slid across with this stroke like that, power stroke, you're going to put it back into position, it'll slide back like that. So here it's saying what the uses of ATP are in all of those steps. I've just said them there. So the ATP is needed for, number one, is breaking the actin-myosin cross-bridge. Just being lazy and writing it short for the sake of time. As well as that, it's needed to, to reset myosin head position. And then it's also needed for active transport of Ca2 plus into the sarcoplasmic reticulum, SR, when it's all done and dusted. Now it says, during late pregnancy, women find ventilation more difficult as the developing fetus reduces the volume of the thorax. This can lead to tiredness and difficulty breathing. A student used the spirometer to measure the ventilation of a woman who was 36 weeks pregnant. So again, they're just measuring the time and how much volume of air they're moving, right? So this is a spirometer. Volume of air. So they're breathing. When you use a spirometer, you put a tube around your mouth, you breathe in and out. So if the volume of air is going up, this means they're breathing out, in, and so forth, right? It says here, mean oxygen uptake at rest is about this. And it says, using these data, the student made this conclusion. They said that being pregnant reduces the rate of oxygen uptake by 20%. Evaluate this claim using the data there. So the mean oxygen uptake, remember, is 0.020 dm3s minus 1. Okay, so the oxygen consumption, or the oxygen, in this case, uptake, is that what they call it? Yeah, the oxygen uptake is the decrease in the volume of the gas in the spirometry chamber. And what you do is you just look at the average slope of the trace across the entire thing, usually. So, what I mean by that is if I draw a straight line from here to here, and then at the very end, of the last point, if I do there to there, right, for 4.5, and then we end at, what, like 3.1, let's go? So then the change in our volume is going to be 4.5 minus 3.1, which if I do that, 4.5 minus, I get 1.4 dm cubed. And over what time frame this happened was 80 seconds. So then the uptake in decimeters cubed per second would be 1.4 divided by 80, so 1.4 divided by 80, which gives you 0.0175. So then mine that I got is 0.0175, and the student says that my data shows that being pregnant reduces the rate of uptake with this, using this. So when you're evaluating, the rules are, you've got to look at this and say, can I say that this is 100% true and I can apply it to absolutely everyone in the universe? So what you should always do is say stuff that supports this, so what it is and what it means. So like you see a pattern that matches theirs, and you say, oh, this is good because, and then describe what you see and then explain why it fits. Then, what else? Contradicting is what you see that's going against it and why that could be. And then validity as well. So validity means, like, can you take this and apply it to every single other scenario in the universe? If they got the same other woman and did it again with, like someone else, same woman did it again, would it be the same? If they got a different woman did it again, would it be the same? Can I take this and apply it to absolutely every single person in the entire universe? So, for example, if I give you chocolate, if I come up to you and I say, yo, bro, I gave... 10 people in a room a banana, and all 10 people shat themselves. And then I said 100% of people must shit themselves. You would be like, what are you saying? That's wrong. Because then you'd be like, you've only tested 10 people. Yeah, fine, okay, 100% of yours shit themselves. But like, what about this and that? And you're already doing it, aren't you? You're literally doing those exact things. So here as well, you would describe and explain what supports it, what's against it, and so forth. So here, the supporting claim is, well, let's have a look at what they're saying. It reduces it by 20%. So if I calculate my one, it's going to be 0.0175 minus 0.02, or the other way around really, because that's going to be a negative number, but it don't matter, 0.02. If I do that and then do 100 times that, I should get 12.5%. So they're saying it's by up to that, so supportive. I mean, in this case, the decrease was 12.5%, which exactly fits them, right? So therefore, they may be correct. However, is there anything that's going against it? Not really in this case, because the exact thing is what we just calculated. So we don't really have much on contradicting. But then let's look at validity, right? Can I take this and say it to absolutely every single woman in the entire universe? So let's go look at this. So then here it says, blah, 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 blah. A student used a spirometer to measure the ventilation in a woman who was 36 weeks pregnant. Already, they did one woman, and they're saying being pregnant does it. Like, throughout pregnancy, you're not going to be the exact same, right? So validity, already I can say that, well, it's only one woman. But as well as that, you know, it was only 36 weeks. Doesn't represent the whole pregnancy, because that's what they're saying. They're saying, oh, a pregnancy does this. As well as that, like, that's not very short. Like, maybe you could have gone even lower, or maybe you could have stayed the same. Maybe you could have gone, okay, but like, you know what I mean? Like, it's only 80 seconds as well. That's a short time. So I'm not happy until you do that again and get some better results, lad, because I'm not going to go and do that with everyone else. It's kind of what you got to think almost. Chromista are photosynthetic protoctists that live in water. Chromista are different from other photosynthetic organisms because they have chlorophyll C, which is not found in plants. Outline the importance of photosynthetic pigments in photosynthesis. So what you need to think is what the point of these pigments are, what their purposes are. What they do is they will absorb different wavelengths of light, right, which then excites electrons, right, and then that's not enough. You need to say where it's going, what's happening. In light-dependent reaction, electron is absorbed, and the electron absorbs light energy, and then what it does is it gets really excited, like woohoo, and then it goes down the electron transport chain, blah blah blah, and its energy is used to pump protons into the stroma, and so forth and whatever, right? So then we need to talk about that. So then the electron is released, is passed down the electron transport chain, it's passed on to NADP for the light-dependent reaction. As well as that, photophosphorylation takes place to replace that, as well as what you can also. And the whole point of that is to make ATP and reduced NADP for the light-independent reaction, or Kelvin cycle. The wavelengths of the light absorbed by chlorophyll C are different from those of A and B, suggest why it needs pigments that are different from those that of other photosynthetic organisms. So when you're looking at an object, for example, right, and it's green, what's happening is green light is going into your eye. So if you put white light towards it, white is all colors. That means that this has absorbed everything except green and reflected it back. So if they've told you that this has got different wavelengths than chlorophyll A and B, it clearly means that it's got, you know, different wavelengths that it absorbs. The next step, though, is to think about what wavelengths you would rather have. So we need to think about where they're living. They're living in water, right? Which means that the water's probably going to lean more towards the bluey side of the colors. So when you've got the electromagnetic spectrum, red is like your longer wavelengths, blue is your shorter ones. So it's going for more bluey light, it's got shorter wavelengths. So they need to absorb light of shorter wavelengths. Because we need to think, why are they only going for those ones? It must be that the other wavelengths don't reach them in deep water. It's the only explanation they can match that. Figure 17.1 is a chloroplast found in the chromista cells. You got thylakoid stacked in trees. You got an external membrane, and they've got one lovely DNA there. And it says outline the structural differences between the chromista chloroplast and the chloroplast found in flowering plants. Already I can see that it's got fewer thylakoids. It's only got like, you know, a couple, and they're only stacked in 3, so nah. And as well as that, there's no connections between the two of these. There's no, you know, lamellae between them, you know. And also like plants, because we're contrasting them, remember, have more than 3 stacks, you know, or groups of that. And also there's no starch grains. So there you go. Cool. Figure 17.2 is the diagram of a plasma membrane of the chromista cell. State and explain how one property of the region A contributes to the stability of the plasma membrane. So it's, what is it pointing at? It is pointing at the hydrophobic region as fatty acid tails. So it's, you're going to point out, aren't you? You're going to say what it is and what it does. It's hydrophobic. That's its property. And why is that helpful? Well, you could, I don't know, you could talk about it's an explanation that's relevant to it. So in this case, what it's doing is that it's got a middle bilayer there. So what it's doing is it's separating 2 aqueous regions. It's got this nice little barrier to the water out there and the water inside it. But as well as that, you could also say this thing has cholesterol in it, which can control the fluidity as well. More cholesterol, less fluid. There are differences between the plasma membrane and membranes within cells. Outline the role of membranes within cells. So we've got a cell, we've got an outermost membrane, and then, like, for example, your chloroplast has its own membrane, and your mitochondria also has its own membranes too. So you need to think, why on earth would I want my own membrane in my own organelle? Why do we have a membrane for other stuff? It's to, you know, stop things leaking out. It's to separate it from everything else. It's to stop things getting in and out that we don't want to. So in the same sense here, that's probably what's happening with these. So when you've got these organelles like mitochondria and the nuclei and this and that and thingamabobs, it's for that exact reason. So what it does is it separates the organelle, like organelles for example, into compartments. But you need to say why that's important. So if I take, for example, the mitochondria, so for example, it's important because chemical reactions happen there, for example, such as oxidative phosphorylation in mitochondria, right? And that's quite important because, for example, if you start leaking all those enzymes out and stuff, it can actually cause damage to cells as well, and we kind of don't really want to do that. Other things you could say as well is that it allows attachment sites for like enzymes and stuff as well, so ATP synthase, for example, but like that's the one that's screaming out to me, you know? Plants lose water by transpiration. As I've said before, transpiration is where water evaporates here and then it's lost through the stomata of the leaves. The rate of transpiration varies between different species of plants. The rate of transpiration can be measured using a pedometer. Plan an investigation into the rate of transpiration in 2 species of plant that would allow the valid data to be collected. Valid means that you can take this and apply it to absolutely every single one of these 2 species if you did it again and again and again and again and so forth, right? Whenever you're doing this, rule number 1 is to identify, first of all, what your dependent and your independent variables are. And then as you go along, you're thinking about what you're controlling that could screw up those independent or dependent variables. So independent is what you're changing, which is going to be our plant species, and our dependent is going to be the rate of transpiration. But we can't just go ahead and say, oh, rate of transpiration. How are we actually going to measure that? In this case, using a pedometer, it's going to be the distance moved by a bubble per unit time, because it's a rate. Okay, so it's very tempting to be like, oh, you know, rate of transpiration, but like that doesn't help you figure out what's going to screw it up, if that makes sense. Because then when you're writing your method, you need to, at each step, think, is there anything that could go wrong here that would make this go higher or lower or mess it up? And then it gets into place quite nicely. So then what you need to do is, the methods, what you would do is you would just, set up the potometer, kind of skip through that just for the sake of time, blah, 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 right? And then your variables, while you're setting up, you're thinking, what could screw this up? So some variables you can think about is we're only changing the species, so we want to keep the temperature consistent, because if it's higher, that can make transpiration go faster, as well as that humidity, as well as that wind movement or like air movement, as well as that light. You want to keep that consistent, so stomata not opening, as well as that it's the surface area of the leaves, because if you've got more stomata, that's not going to be a good thing, as well as that the plants themselves, like the age of the species as well, and then as well as that, you know, for example, resetting it as well, so resetting with the reservoir. Because a pedometer is basically a big long tube, blah blah blah, and then there's this and whatever, blah blah blah, and there's this leaf here, and you see a bubble and you measure it by moving it across like this, and then you have a tap here with some water that you put in and it resets the bubble, right? So it's just thinking about what can screw those things up, blah blah blah. So I'm just kind of skipping through that because you can look at your required practical notes for that. It's just probably not practical for me to sit and yap about it for that long. And also I'm trying to rush through these as quick as I can. So we've done, let's say we've run that, right? But then we also want to make sure it's valid, meaning you can do it again and again and again. So if I do it once and I get one, like, let's say for example I weigh, I don't know, a block of cheese and I get 100 grams, and then I do it again and I get 50, and it's in the same bag. Does that mean that every single one's going to be these 2? We don't know. It's not valid. If I do loads and loads and loads, there's more that's 50, 50, 50, 50, then I'm like, okay, right, okay, now it's looking more like it's 50. First of all, let's talk about how we actually measure the rate. So you would measure the distance, where am I writing up there? Sorry. Distance moved by bubble, right? And then what you would do is also the time, like, you know, I don't know, like every 10 seconds or something like that, so, you know, continuously if you can. And then what you would do is you would calculate the rate by doing, you know, like for example the movement of the bubble per time, so whatever it is divided by the time it took, or it could be the volume, so you could do like the you could do the volume of the thing by assuming it's like a sphere moving, like a cylinder moving across or something like that, whatever you choose, so a movement whatever, right, over time. And we're obviously going to repeat it of the 2 species, but then like I said before, you don't want to just, you know, do it and then be like, oh yeah, that's one done. You want to get repeats for replicates, and then you want to calculate a mean. And we're looking at how, if the species, changes the distance of the movement of the bubble. So you're going to compare the means to see if there's a difference between them. So the statistical test you would do is a t-test to compare the differences in the means to see if one's significantly higher or lower or whatever, right? So sequentially, you describe the method, and at each step you would say, when doing this, control this or whatever, like I've written here, and then you would say how you do it and all this stuff. Just have a look at what you may call it, your notes, because it's not really too essential to talk all about how to set it up and this and that, other than when you're controlling this, obviously. Plant cell walls are made of cellulose, and it's a polymer of beta-glucose. Give 3 properties that make it useful. So I mean, number 1 is it's insoluble. You don't really want your cell wall to just dissolve. That's probably not a good idea. It's got a high tensile strength. You can't just sit and say rigid. What that means is that it can resist being pulled. As well as that, it is flexible, and as well as that, you can form hydrogen bonds with neighboring chains as well. So there's a couple. Now, cellulose cannot be digested by animals. Some animals have bacteria in the stomachs that produce enzymes that can digest cellulose. Explain whether the actions of these is intracellular or extracellular. It's told you that some mammals have bacteria in their stomachs that can digest it. So the bacteria are doing the digesting. And it's saying, is it intra or extra? Now, cellulose, we just established, is a polymer. And this is a bacteria that's like one cell. It ain't going to fit. So it's got to be extracellular. Why? Because it cannot enter the cell. Like, imagine you got this gigantic cell wall, loads and loads and loads. You got this teeny bacteria. It's like it can't get in. So what it does, it will digest it by you're using these enzymes on the outside and then absorb the beta glucose in instead, like that. Honeypot ants belong to several different genera. Some specialized individuals are used as food storage individuals. These individuals have swollen abdomens that store various foods, which can be given to members of the colony when required. So one such individual is here. It says, an investigation was carried out into the respiratory substrate of 3 different genera of honeypot ant by measuring oxygen uptake and CO2 production. So again, we're looking at our dependent and our independent. So dependent, independent, what are they changing? It's the genus. And then what are they dependenting, or what they're measuring? CO2 produced and O2 consumed. So there's 2 of them. Here then, it says, use this data to suggest the likely diet of each genus of honeypot ant. So then it says, justify your answer. So here, to me in this table, because they've given me CO2 produced and O2 consumed, and they're asking me what they're using as their diet, it's screaming to me that we're probably going to have to work out the respiratory quotient for each of these. The respiratory quotient is your CO2 released, its volume, over its O2 consumed. So we're going to work out its respiratory quotient for each of these. If I just rapid fire that, that's like nearly 1 already. If I do the other one, it's going to be what? So 0.559 over 0.66, which is what? 0.89, which is roughly, yeah, we'll keep it 0.89. And then here 1.01 divided by 1.47 gives us what? 0.68. Something. So if I just round those up, that would give us 0.9 and 0.7. So as well as that, we need to now look at the respiratory quotients of different things and figure out which ones these fit. So respiratory quotients, you can use they're used in other substrates in respiration, and anything that's lower than 1 is going to be a lipid or a protein. The reason being is because you need to put more oxygen in to oxidize it versus like lipids and proteins rather than carbohydrates. Because this is the CO2 over the O2 in, right? CO2 out, O2 in. If it's 1, it means roughly equal amounts, but for the others, you're going to have more O2 than CO2 out, which means that gives you in on what's going on here. So in this case, generally speaking, lipids have the least respiratory quotient. They need the most oxygen in and CO2 out. So that kind of gives it here as well. So in this first one, I mean, camponotus, whatever, it's carbohydrates. Why? Because its RQ is roughly 1. Then in the next one, it's mellophorus. That's 0.9. It's probably going to be proteins. Or it could be a lipid and carbohydrate mix. Could be the 2 of them. Why? Because its RQ is 0.9. And then this one's got the lowest. It's most likely lipids. So it's got to be 0.7. There you go. So chitin is a polysaccharide found in insects. It's used to form the hard outer casing. And here's the structure of it. And it says, using the information, state 2 similarities and 2 differences between the chitin and the glycogen structure. So already off the bat, we can see here, and they've told you in the question, it is a polysaccharide. Right, so if it's a polysaccharide, a similarity by default is that they have glycosidic bonds, more specifically 1,4 glycosidic bonds. Another similarity is that they are polymers. They're multiple repeating units, or polymers or polysaccharides already, right? Because it's saying that between that and glycogen. As well as that, both of them have this side group here as well. And then what else can I see? I think those are like the main ones. Oh, and also, sorry, these are 1, 2, 3, 4, 5, 6. So yeah, they're both 6 carbon compounds as well in their monomers. Because you can see there's 2 repeats of these there. Now, what's the difference? Well, I mean, one thing here is there was nitrogen in there. Like, glycogen doesn't have nitrogens in it. So there's N in chitin, but not in nitrogen. But then as well as that, what we're comparing it's glycogen. Well, there's no branching here. Glycogen can have branching. What else can we see here as well? Well, so glycogen forms branches by forming 1,6 glycosidic bonds as well. So what that means is that like this bit here can form a glycosidic bond with another bit from there, right? So in this case, it can't do that here. There's no evidence of that. So there's no, you could say as well as that, no 1,6 glycosidic bonds. But those are a couple. Now it says, insects use glucose to generate ATP. Outline the processes involved in the generation of ATP through chemiosmosis. So all they want to talk about is the chemiosmosis part. They don't care about anything else. So in that case, if we're talking about this, let's talk about the overall purpose of chemiosmosis. The entire point of chemiosmosis is to proton out of the matrix into the intermembrane space. You create a proton gradient, and then you use that to drive ATP synthesis. If I draw a quick diagram of what's going on, matrix, intermembrane space, right? What happens is we're trying to shove protons out of the matrix like this into here. Once it's in the intermembrane space, obviously the concentration is going to be higher here, so the proton's going to want to move down, facilitated diffusion through ATP synthase, which is a channel protein. As it's doing that, its movement is what's driving ATP synthesis, and yeah, that's pretty much it. So this is the chemiosmosis bit. So what we need to do is we need to describe these steps essentially. So let's have a loop. Let me re-raise that and then turn it into words. So H plus ions are pumped out of mitochondrial matrix into the intermembrane space, right? Then what happens is this creates a concentration gradient higher in the intermembrane space versus the matrix. So then what happens is H plus moves down, concentration gradient via facilitated diffusion through ATP synthase, and this movement drives ATP synthesis from ADP PEI, and there you go. Other bits and bobs you could talk about is mentioning the proton motive force, and obviously going into a bit more detail as well, talking about like stalked particles and Christie, but like this is the main chunk of it, to be honest. So student carried out an investigation to look at the effect of ethanol in the cell permeability membranes in beetroot, and they did these steps. Cut equal sides of beetroot with a cork borer, wash them with running water, put them in 100 centimeters cubed at different concentrations of ethanol. So immediately we're looking at our independent and our dependence. You know how it is. So our independent is what? Concentration of ethanol. Dependent is obviously very tempting to go ahead and say, oh, it's the permeability, right? But we're looking at what we're actually measuring with this. So it's going to be our colorimeter. What did they actually measure with it? The absorbance. Cool. That's their dependent. That's what they're measuring. So they place in this blah, 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 whatever, right? And it says each step in the student relies on assumptions. For each assumption, so like the numbered step in the method that relies on that. So the pigment will only leak into the solution if the membranes are disrupted. So the step that relates to that, whatever step is basically, because what they've done is they've cut beetroot, right? It's going to have bits of membrane in it. If you just shove that straight into water, then you're going to have a bit of, beetroot pigment already there. Whereas we want to try and get rid of it so that anything that is out in there and it starts going darker in color or whatever, is because of the membrane disruption. So that's probably either one of two or three. That's why it's rushing it or placing it. So I'm just going to do three. Then absorbance is proportional to the concentration of pigment. That's got to be this last step because they're measuring a colorimeter to then plot it against the concentration and then see if there's a pattern, any proportionality between them. So that's step 5. And it says the pigment will be released when the beetroot is sliced. So what step factors that into the cutting is that's going to release beetroot. So that step, the reason they're washing it is to remove it, like I said before. So that's got to be 2. Next, it says the student kept the ethanol solutions at constant temperature. State other variables which need to be controlled to ensure this method is valid. So that's what I was saying before, is what's screwing up these things, right? We're measuring the dependent variable. So in order to control things, we need to control whatever could screw up the absorbance of the color, right? So we're looking, absorbance, what can affect absorbance from the beetroot? Look at every single step. So for example, you could talk about the age of the beetroot, the length, the volume of it, where they took it from, the time that they spent washing it. The pH is another one you could talk about, and also you could use the same blank cuvette and a colorimeter as well. So any one of them is fine, because all of those are things that are going to screw up this and make it go higher and lower. It's what you should be looking out for and thinking. Next it says here's their figure. So let's have a look. Absorbance, and they've got concentration of ethanol, and again one by one you can see it's sort of going up and then down, right? The absorbance is going up and then back down. What's 3 criticisms of this? Or I was already looking at this. I mean, there's no unit for ethanol. So no unit on x-axis. As well as that, what else have we got going on here? So 0, 1, 2, 3, 4, where's 6? No 0.6. And also, like, what is this, bro? I don't know what this graph's supposed to tell me. No overall title. There you go. It says, explain how carrying out replicates could improve this investigation. Like, if I measured, like, I don't know, again, like I said before, if I measured, I don't know, I buy, like, a packet of crisps, and I measure a crisp at, like, 1 gram, and the other one's, like, 0.00001 grams. Like, which one's the one that's most likely going to be it, Like, I don't know. So if I do it again and again and again, I get 0.0002 and so forth, then I know at least it's leading towards that. I don't know how much a crisp weighs. So the reason we're doing that is so that we can, again, identify anomalies So we can look at what our results are kind of lying at, and also means we can calculate means, and that's helpful because one, it means we can have like a repeatable test. Because if you're getting similar means and not many anomalies, then it's repeatable. But also it allows for a stats test. You could do a t-test as well, or standard deviation. So that's helpful because it means you can check that it's not due to chance, any differences you do see. The greater blue ringed octopus, this is one of the most venomous of all animals, probably lives in Australia, let's be real. Its bite contains tetrodotoxin, a neurotoxin that can cause paralysis and death within minutes. Its following information has been discovered about it on the effects on nerve cells. So what it does is it binds to the external surface of the voltage-gated sodium ion channels in the axon membrane, and then what it does is it changes the tertiary structure of this channel, and then it cannot open. Using this information, explain how it affects the activity of neurons. So it's saying, how does this lead to whatever problem is here? So we need to say its consequences. So what we're going to say is that Na plus cannot enter, which means as a result you're not going to get no depolarization. And then if that means you're not going to reach the threshold, you're not going to get any action potential. And then, I mean, well, it depends what it is. What's this nerve that it's doing? It's axon membrane. So you're not going to get any neurotransmitter release as well. So that's what you need to do. You need to talk about it step by step. So a common cause of death from this is suffocation, not getting enough oxygen as a result of paralysis of the diaphragms. Explain how paralysis of the diaphragm can lead to suffocation. So a lot of people just yap about all kinds of stuff. It's literally asking you how that gets to there, step by step. So the diaphragm is paralyzed. It doesn't, no contraction, which means it does not. So think about it. When you're breathing in, you're contracting your diaphragm. So it makes the volume of your thorax get bigger. You know, breathe in, your chest goes up and out, right? So no increase in thorax or chest cavity volume or decrease in pressure. So then no air enters lungs down a pressure gradient. That's what's happening. Because if you have a water bottle, for example, and you squeeze it, or it's a can, I guess, if you squeeze it with the lid off, the pressure is now really high in here. The water's like high pressure, so it wants to get out down a pressure gradient. Pressure in here is going to be way higher than outside, so it squeezes out like that through the holes. That's what happens. So in the same sense, we're almost like doing the reverse. It's like you already have a squeezed bottle and all the air's out, and then you're letting it come back in again, so it's pushing, the water goes back in. Like that. So in the same way, the air's doing the same. TTX is also known to reduce the speed of conduction in the Purkinje fibers of the heart. Suggest and explain what this effect would be on the heart. So again, it's just like every other one. What happens when this slow down, this next, right? So if that's the case, then you're going to have slower transmission. So slower impulse transmission from, and then this is from your AV node to the ventricles. So then what's the direct consequence of that? Well, it means if there's going to be less of that, then it's going to be slower ventricular contraction or systole. I'm just going to say because I'm lazy. That means you're going to have a increase in time spent in systole and diastole, which then decreases your heart rate because it's beating slower. So there's your problem. So it's again just step by stepping it, saying this, what's next? There you go. Then mollusks containing this have unmyelinated neurons. Saltatory conduction cannot occur in these. Why is transmission of action potentials slower in this? It's because, so when you have a myelinated neuron, here's your myelin, there's that, right? We've got this bit of unmyelinated patches here, so it jumps from there to there. It's faster than going through the whole thing like this. There's no nodes to jump. That saltation is jumping from bit by bit like that. So then, here you would say there are no nodes of Ranvier. So it means there's a longer distance of, depolarization to occur. So it means the whole axon needs depolarization. There you go. I'm just written depol for short. A scientist used a respirometer to investigate the rate of respiration and photosynthesis in maize in different light intensities. So, whoa, what have they been doing then, eh? So, independent, dependent. Again, it's tempting to just go and say rate of respiration and photosynthesis. That's not very helpful because we want to critique this maybe down the line. So it says the scientist placed these made seedlings in a respirometer kept in the dark for 3 hours. Then the respirometer had soda lime to remove any CO2 made from the seedlings. They then put 10 of them in a respirometer without soda lime and then put them in different light intensities at a time. So what their dependent is what they've changed. So what have they actually changed? It was the lux of the light intensity here. And then what did they actually measure as their dependent was the distance moved in the respirometer. Cool. Then just have a quick look at the data, what's going on. So they've increased the light intensity, and we can see that the distance moved by this respirometer was initially negative, and then it's becoming more and more positive. Cool. The diameter of the capillary tubing was this. The volume of a cylinder can be calculated with that. Calculate the rate of oxygen uptake in the dark and give your answer to two significant figures and show you working. So if it's in the dark, there's 0 light intensity, right? And the distance moved by the fluid, so if I imagine this like a tube, the distance here is 3.7. They've told you the volume of a cylinder is this, pi r squared L. They've told you that the, diameter is this. So list out what you have. So the diameter is going to be this. So the R is going to be 0.05 millimeters. What unit do they want it in? Millimeters cubed per hour, right? And this is measured in millimeters. So its length is 3.7. So then pi times 0.05 squared times the length, 3.7, will give us our volume of it, right? So if I do that, I get pi times 0.05 squared times 3.7, which is 0.0202290597. We're not done yet because it wants it per hour, right? In this case, this is the distance moved by it, but we haven't got any time frame, have we? Unless you read the question carefully, because it tells you that it's been left in the dark for 3 hours. So if they want it for 1 hour, this is for 3 hours, you've got to divide it by 3. So if you divide it by 3, then you end up getting, this gives you 9.67 times 10 to the minus 3. And it wants it to two significant figures there, so that's going to be 9.7 times 10 to the minus 3. Cool. Here it says 1,700 lux is a typical light intensity on a cloudy day. Calculate the percentage increase in gas production between these and then show your working. So what we need to do is we need to find out the difference in the gases between these two. So if we look, we've got here 2.9 and then we've got 1.2. So the difference between these two is going to be our increase. So 2.9 minus 1.2, but they want the percentage increase. So it's going to be this divided by 1.2 times 100. If we do that, we should get 141.67, right? But they've given us all of these numbers to two significant figures, so we are also going to follow, so that's 140. So just why the soda line was not put in the respirometer when you grew them in the light. So we need to think about what's happening here then. So let's have a look, shall we? We're looking at respirometer. If I rub this out, if they got it in the light and whatever, blah, blah, but then they did 10 of them in separate and put them in different light intensities. So then, so this absorbs CO2, right? And they've grown it in the light. If we took CO2 out, then we would not get any photosynthesis. No CO2, no photosynthesis. Which would not be very useful, considering they're looking at the rate of respiration and the rate of photosynthesis in different light intensities. How are they supposed to measure if there's no CO2 there? Then it says the scientists made this claim. The results suggest that in maize seedlings, the rate of photosynthesis only exceeds the rate of respiration when the light intensity is above this. Use the data in table 2 to explain why they did this. So if we look at 1510, which is here, the distance moved in this respirometer is 0. Now think about it logically for a second. In this respirometer, what's going to happen is they're going to respire to put out CO2, right? These little seeds. If they put out CO2, and this is in the light, right? There's not any soda in there, so there's not going to be absorbed. So the CO2 is chilling here, but then they're going to use it again in photosynthesis. So CO2 is out, but then it goes right back in for photosynthesis. But then it's constantly cycling like that. So if there's more, if there's an equal amount in and out, because again, these are in light, so the CO2 is not permanently disappearing, then they're equal. So if you have a higher, right? So if you've got a higher light intensity, then it means you see the distance moved by is now positive, which means that you're now outdoing photosynthesis versus respiration. So in this case, that's what we'd write. So the distance moved is 0, therefore photosynthesis rate equals the resp rate. So there's no net or overall change in air volume, because everything they're putting out is going straight back in. So the student looked at slides of different tissues under a microscope. The 4 tissues are labeled W, X, Y, and Z. What are W, X, and Y? So if we have a quick look, shall we? What have we got going on here? So if you kind of follow this around, you can kind of see there's these tubes all branching out here, right? Feeding into this one bigger one here. That kind of looks awfully like liver tissue with your central vein and, your canaliculi and all of that kind of stuff. So W is most likely going to be the liver. Alright, now they want X. So if you look at X, what's going on here? So here we have like these clusters of like paler tissues, and paler cells in amongst all of these sort of bright, darker looking ones. I'm kind of like, And within these paler tissues, I can see darker and lighter stained things. The darker ones are beta cells, and the pinker ones are alpha. So to me, this is pancreatic tissue. Next, they want to know what was the last one. It was Y. Already, I can see those lovely, lovely striations. You can see there's multiple nuclei as well. You can see there's lovely alternating iron A bands. This is skeletal muscle. Pancreas or pancreatic and skeletal muscle. Cool. So the student wrote the following summary about the control of heart rate. When the heart rate is too low, the level of carboxylic acid in the blood becomes higher than normal. The vagus nerve sends action potentials to the AVN to increase the contraction rate of the heart muscle. The baroreceptors in the walls of the blood vessels then detect that and so if it is normal, heart rate return to resting. The endocrine system also changes the heart rate, releases the hormone adrenaline from the adrenal medulla, causes smooth muscle heart. I actually want to. That makes me feel sick. So, real quick, okay. All nervous things and a lot of hormonal things follow a similar pattern of a receptor is detecting a change of some sort in your internal or external environment. It's detecting a stimulus. Something that is specific to that. So, for example, baroreceptors for pressure. What it will do is it will send an impulse across the sensory neuron to the central nervous system or some sort of bit that's processing it, right? So in this context, it would be the medulla. And then what it will do is it will send, impulses through either the sympathetic or the parasympathetic nervous system or through like motor neurons to skeletal muscle or whatever to an effector, which then changes its behavior depending on what. Right? So in this case, they've said that when the heart rate is too low, this carboxylic acid becomes higher than normal. I mean, already it's not carboxylic acid. That's just a general class of them. It is carbonic acid instead. Then it says the vagus nerve sends action potentials to increase that. The vagus nerve is parasympathetic. It should be the accelerans nerve, or you could say it's a sympathetic one. Here as well, it says it's to the AVN. The AVN is not the effector here. It is the SAN, because the SAN sends impulses and is the reason the heartbeat is starting to begin with, like it's doing its job. So then if it's firing faster, it means that it's going to give out more impulses to reach the AVN and blah 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 so forth, right? It's the thing that's starting it here. Then what else have we got? To increase it. Then the baroreceptors in the walls of the blood vessels then detect that the pH is normal. Hello? If it's baroreceptors, that's going to be pressure, or if it's this, then it's got to be chemoreceptors. So the receptors here are chemoreceptors. And then as well as that, from the adrenal muscle to cause for adrenal medulla to produce smooth muscle, it's cardiac muscle. Different type of muscle there, mate. So there you go. Reflex actions are rapid responses to protect the baby from harm. The moral reflex is found up to 5 months of age and occurs when the baby feels like its head is suddenly no longer supported, and it's made up of the following responses. So what they do is they spread out their arms and bring them closely together, and the baby cries. This is a real thing, and that does happen. We check for it with new babies when they're born to make sure there's no problems in that sense. So it says, suggest how the moral reflex helps prevent harm from a newborn baby. It spreads out its arms and brings them rapidly together, and it's crying. How does this stop harm? Well, number one is that when they're doing that and they're putting them together, they're trying to, hold on to something, which could be like a tree branch or something from back in the day. I don't know what we were doing back in the day, right? But there's that. And then as well as that, why would crying help? It brings attention to them, to the baby. But yeah, if there's problems with that, it can suggest there's, things going on, maybe just to do it right. But it can also be things like neurological damage and all kinds of bits and bobs. But yeah, and also they can come back as well if things like that happen. Oh, and that's actually what's going on here. It says the moral reflex gradually disappears and then stops completely after babies reach 9 months, and then others develop as children grow older. Describe a reflex response of a 3-year-old child would make to an object moving towards their eyes and explain the advantage of this response. So a reflex is something that you do not think about, it just happens. It goes straight in the spinal cord and back out, and it doesn't have to go to the conscious parts of your brain. And obviously in the head it's a bit different, but like, whatever, right? So here, if think of a 3-year-old and an object moving quickly towards its eyes, what's it going to do? It's going to blink. They're going to blink. Right? Baby's gonna blink, or the kid's gonna blink. Why is that helpful? Well, it's an involuntary response, and it's unconscious, which is faster. And what might that be useful for? It prevents damage to the eyes. Like, if I said I put my hand on a hot plate and I say, why is that important that I do that when I move it really fast? You're gonna be like, oh, so it doesn't burn your hand. It's the same thing here, right? There you go. Hope that helps. Thank you very much. Please like, share, and subscribe. You know the drill. And yeah, I'll carry on.